In this short video, I am going to discuss the Transfection Q-labels protocol, which is part of the cell biology protocol collection. Protocol is designed to measure the proportion of fluorescently labeled cells or nuclei expressing a fluorescently labeled recombinant protein. As shown in this image, we have nuclei which are labeled with DAPI and our transfection marker in Alexa 633. I'm going to close this image. To load the protocol, select the count size ribbon and then press the protocols button. Selecting the cell biology collection, scroll down until you reach the Transfection Q-labels protocol. Simply press the load button to load the protocol. All protocols include a demo image or demo image set. You may also use your own images for the protocol. To load the demo image, select the drop down next to the reset button, selecting the load demo with the open demo image checked. Next, press the sync button to load the image channels. You will need to do this for your own sample. In this case, the cells are labeled with DAPI, and the label, the transfection label, is Alexa Fluor 633. To test the settings of the protocol, make sure the interactive option is checked, and press the next step button. The nuclei are segmented using the histogram segmentation, which can easily be adjusted. In addition, you may also use the smart segmentation machine learning segmentation method, whatever is appropriate for your sample. When you're satisfied with the segmentation, simply press the OK button. The protocol includes a few segmentation options. The first one is minimum area, which you can use for filtering out small fluorescent areas, maybe cells, debris, and you can use this to filter out high and low based upon the area. Next, there's a clean borders option. The default protocol has all borders turned on, so you're not counting any nuclei touching the edge of the image frame. Fill holes. If there are any gaps in the segmented nuclei, they will be filled. And finally, object splitting. There's a choice of none, watershed, or boundary shape. Right now, boundary shape is on by default. If you make any changes to the segmentation option settings, press the apply button. Next, press the next step button, and now we'll segment the Alexa Fluor 633 channel, which is the transfection channel. In this case, the nuclei are already segment. However, we can improve the segmentation a little bit by looking to see if there are any other cells that were missed. In this case, we can now draw additional reference objects. And I'll update the background. And now we can see we've picked up a few other nuclei. As always with smart segmentation machine learning, you can draw additional reference objects to find your transfected cells or additional background objects. If you need to make further adjustments, you can always press the clear button and draw new reference objects or create your own recipe. Now the transfected cells have been identified, press the OK button. This step of the protocol also includes similar segmentation options of minimum area, fill holes, and object splitting. Here, we're specifically looking for fluorescence in the outline of the nuclei, which are outlined in yellow. Press the next step button, and now the calculations have been made. In this case, we have 137 cells. 11 are transfected, for a transfection percentage of 8%. If you've made changes to the protocol, wish to save those for further use, simply press the Save As button. This will create a new protocol saved under the Transfection Q-labels protocol group. To continue, press the Finish button. And now if you'd like to measure additional images, if they're open on a desktop, you can select the next image and press the next step in the interactive mode. For batch processing, if you have multi-well plate data or a folder of images, simply uncheck the interactive. If your data is in multi-well plate format, simply select the rows or columns you wish to analyze and make those active, and then run selected. Or if your images are in a folder, simply select the Run Batch and point to that directory of images. To export the data, simply press the Excel button.